Hi, Dr. Manella. So in this video, I'm going to explain 8 major points of what you've taught this semester. I'm going to start with DNA, because that was the first topic, and where else would you start other than the most fundamental part of all life? DNA is in the nucleus of eukaryotic cells and is organized and packaged into chromosomes. In prokaryotic cells, it's a singular circular strand that's just supercoiled for condensing, so it's not as complex as eukaryotic cells. In eukaryotic cells, this process seems simple, but our DNA is very long, and the nucleus is not that large. The DNA has to be condensed while also being accessible for DNA replication and other functions at all times. This condensing is done in levels. The first level of condensing has nucleosomes. Nucleosomes consist of 4 histone proteins, H2A, H2B, H3, and H4. They're found down here. There are 2 sets that come together and make a histone octamer. It's like a spool, and DNA wraps around the octamer about 2 times, as seen here. To keep the DNA on the histone, there are histone tails. Histone tails are rich in lysine, which gives them a positive charge. The positively charged tails keep the negatively charged DNA in place. This looks like a pearl necklace with linker DNA connecting each nucleosome. The string of nucleosomes is then coiled by the H1 histone protein. The H1 protein changes the path of the fiber by attaching where the DNA exits the nucleosome. This coil of the nucleosome string creates a 30 nanometer fiber. From the 30 nanometer fiber, there isn't much known about the organization, but the fiber is bunched, looped, coiled, and poof, there's a chromosome. Although we don't understand much about the organization of chromosomes past the 30 nanometer fiber, we do know about the shape of the DNA during different parts of the cell cycle. During interphase, DNA is replicated, so chromosomes are at their least condensed state. Unlike interphase, during mitosis, chromosomes are highly compacted. This is because during mitosis, the cell has to distribute the homologous pairs of chromosomes to each daughter cell. The process of going from very compact chromosomes in mitosis to a less condensed state in interphase has to do with regulating chromatin structure. If chromatin is tightened, then the DNA is inaccessible, but when it's loose, then it's accessible. One of the ways this is done is the chromatin remodeling complex. Chromatin remodeling complexes work with hats and H-stacks to tighten and loosen DNA around the histone. Hats stand for histone acetyltransferase, and H-stacks stand for histone deacetylase. The histones have amino terminal ends or tails, and these tails are rich in lysine, which I stated earlier, giving them a positive charge. When the hat comes in, it acetylates the histones, giving them a neutral charge. Now that the histone tail is neutral, the DNA is loosened. The histone remodeling complex unwinds the DNA while also threading the linker DNA onto the histone octamer. When the histone is in place and the DNA needs to be tightened again, H-stacks comes in and deacetylates the histone tail, returning the positive charge. Now that the organization and compaction of DNA is understood, the next thing to look at is how DNA is replicated. DNA replication is semi-conservative, so one strand is old and one strand is new. DNA replication starts with breaking hydrogen bonds, which is not an easy task. Cells have initiator proteins that break the hydrogen bonds starting at the replication origins. Human DNA has many replication origins or replication bubbles. This makes DNA replication much faster than if it had to start at the beginning and go all the way to the end. From these replication origins, there is a replication fork. Those replication forks are bidirectional, so they move in separate ways until all of the origins of replication meet. As you can see here, that's what's happening. These are the origins of replication, and they're moving bidirectionally, getting larger, until the replication bubbles eventually meet and create two double-stranded DNAs. The enzyme responsible for pulling DNA apart is DNA helicase. DNA helicase unzips the DNA, and then single-stranded binding proteins bind to the unzipped strand of DNA to prevent them from rejoining. The fork is anti-parallel, and DNA replication can only happen 5' to 3'. Since the fork is anti-parallel, one of the strands will be fine and flow normally, but one strand will be facing the wrong way. The DNA will have to be made starting at the 3' end, which takes longer, coining the name the lagging strand. The strand made continuously starting at the 5' end is called the leading strand. RNA polymerase, called primase, starts the replication by adding an RNA primer at the 3' end of the template strand of DNA. DNA polymerase then connects to the RNA primer because RNA polymerase can make DNA from scratch, while DNA polymerase needs other nucleotides to add to. DNA polymerase makes a phosphodiester bond between the new base and the existing base. The DNA polymerase uses bases called deoxynucleotide triphosphates. These DNTPs bring their own energy to the DNA polymerase. While the DNA polymerase is on the DNA, it doesn't want to stay, so a clamp protein is attached to keep the DNA polymerase on the DNA. The clamp protein was put on by a clamp loader, opening the clamp and then threading the DNA through. The lagging strand is made in Okazaki fragments, which is the same process as the leading strand, but broken up. Each fragment starts with RNA primase and a new DNA polymerase and clamp protein. The Okazaki fragments are joined through 3 steps. An enzyme chews the RNA primer away, and repair polymerase fills in the missing DNA. Then DNA ligase comes in and joins the backbone together. The only problem with this process is the RNA primer at the end of the strands would keep getting smaller and smaller because DNA polymerase needs something to land on to fill in the missing DNA from the enzyme eating the RNA primer. The solution to this is telomeres. Telomeres are added to elongate the ends so important DNA isn't lost when the strands are shortened. As DNA polymerase moves along adding bases, it proofreads as it goes. It has this thumb-like structure that feels the back of the DNA to check to see if it's smooth. If it's not smooth, then there was a mistake, so it backs up to fix it. That's seen here as the ribosome is editing because there was a mistake. Whenever mistakes do slip through, though, they could be very detrimental. DNA mismatch repair comes into play if a mistake slips through. DNA mismatch repair recognizes mistakes by feeling the DNA backbone for anywhere that isn't smooth. In bacteria cells, the mechanism looks for the methylated strand. The methyl groups are attached to the old strand of DNA, so the repair enzyme knows to fix the non-methylated strand because it's newer. This is not how it works in eukaryotes. We don't know how the cell knows which is the old strand, but it must, because DNA mismatch repair works in the same way. DNA mismatch repair works by DNA nuclease removing a section of the damaged DNA, then repair polymerase comes in and fills in the missing DNA. Finally, DNA ligase seals the backbone together. Another type of mutation that the cell must fix is a double-strand break. Without fixing a double-strand break, the cell would die. With this in mind, there are two ways to fix a double-strand break: homologous recombination and non-homologous recombination. Homologous recombination is complicated but elegant, while non-homologous recombination is quick and dirty. For non-homologous end joining, the break is chewed back to make a blunt end, and then those two blunt ends are glued together. This is efficient because it fixes the break quickly, but any genetic information in the sequence that was chewed back is destroyed, and this can silence genes for whatever might have been encoded. Another mechanism that saves the DNA sequence is homologous joining. Homologous joining starts with resection. The 5' end is resected for a really long 3' overlay. This overlay nuzzles and tests the strand until it finds a complementary base. This complementary base comes from the homologous chromosomes, so if the break is in the chromosome your father gave, your chromosome from your mother is intact and can be used as the template. When the 3' overlay finds a complementary strand, the top strand lifts up and creates a D-loop. The top of the D-loop is used for the template at the top, and the bottom strand is used to connect the bottom. This looks like DNA crossing over. This crossing over creates two holiday junctions. The holiday junctions are snipped and then glued to the DNA. Now there are two strands of DNA and none of the genes were silenced. This is one of the leading factors for evolution. This mutation can be used to drive evolution. Sometimes the cell will purposely cleave its DNA and cause a double-strand break, so it will have more genetic diversity. That is the process of replication, and some of the repair mechanisms associated with replication. Now we will look at the central dogma. Starting with transcription. In eukaryotes and archaea, there is a pre-initiation complex that forms before transcription can occur. There is a Tata box as a promoter sequence on DNA. Transcription factor 2D has a small subunit called Tata binding protein, or TBP. The TBP binds to the Tata box and bends the DNA at a 90-degree angle. Once transcription factor 2D, or TF2D, and TBP bind to the Tata box, TF2A is recruited, and then TF2B. Once transcription factor 2B is bound, RNA polymerase II binds with a small subunit of TF2F. TF2E joins the complex, and finally, TF2H binds to the tail of the ribosome and completes the pre-initiation complex, allowing transcription to begin. The TF2H has many different functions, like having the helicase, along with it interacts with the mRNA as it leaves the ribosome. The entire transcription process happens in the nucleus, so once the RNA is made, it needs to be processed before it can leave the nucleus. The TF2H puts a methylated guanine cap on the mRNA. It attaches the cap upside down so the 5' end is on the 5' end. Next, a long chain of adenine bases are added to the end of the RNA. These two things tell the cell that this is a whole messenger RNA. If there isn't a tail or a cap, then the mRNA is broken, and the cell doesn't want to make a protein from a broken mRNA. This happens after the RNA goes through another process to remove introns. Introns are non-coding DNA, and exons are coded DNA. To remove the introns, spliceosomes splice the intron and glue the exons together. There is alternative splicing where some of the exons can be removed at the same time as the introns, which makes a new type of protein. When RNA is completed, the guanine cap reacts with the poly-A tail. So it does this. It creates a circle, and then the guanine cap binds with the poly-A tail. Creating a circular mRNA that leaves the nucleus. The process of making proteins is translation. Translation starts with the mRNA and is decoded in sets of 3 nucleotides. 3 RNA bases are called a codon, and each codon codes for a single amino acid. The genetic code is like a Rosetta Stone for the amino acids. There are 64 possible combinations, but only 20 amino acids. So many codons code for the same amino acid. This redundancy allows for a buffer if there is a mutation. Maybe the mutation encodes for the same amino acid as the original sequence, which will make the same proteins. This is a silent mutation. Translation starts with the AUG codon. This starts the reading frame for the ribosome. For every codon, there is a tRNA with an anticodon to pair with the codon. The tRNA is charged with an amino acid by aminoacetyl tRNA synthase. Once the tRNA is charged, they are ready for translation. But they need ribosomes to know when and where to bind to the complementary bases on the mRNA. Ribosomes are made of 2 parts, a large subunit and a small subunit. In bacteria cells, the small subunit has an RNA attached to it with a Stein-Delgarno sequence that searches the mRNA for the AUG start codon to initiate translation. In eukaryotic cells, there is an initiation factor that binds to the 5' cap on the mRNA. The mRNA is threaded in between the 2 subunits. There are 3 voids in the ribosome that allow for tRNA to bind. First, tRNA binds to the A spot. Then the ribosome moves forward, so the tRNA in the A spot moves to the P spot, because the tRNA stays in the same spot while the RNA moves forward. When a new tRNA moves into the A spot, a peptide bond is made between the amino acid of the tRNA in spot P and the amino acid of the tRNA in spot A. The peptide bonds go to the amino acid in spot A. the ribosome moves forward, and the tRNA from spot P moves to spot E because it no longer has a peptide chain, and the amino acid from spot A moves to spot P because it has more than one peptide. This continues until the ribosome gets to a UAG sequence. It doesn't encode for an amino acid, but instead a release factor protein. When the peptide chain tries to make a bond with the protein, it leaves the tRNA but can't bind, so it's released. The small subunit leaves, then the large subunit leaves, and translation is done. This process of replication and transcription and translation are incredibly intricate and amazing, but being able to do this and having a genome isn't enough. Gene regulation is what determines when and which proteins we make. It allows neurons to be made in the brain and not in the liver. Every cell has the entire genome, but there's no reason it needs to be used. Gene regulation works by regulating the genes expressed with proteins and signals. Combinatorial control is one way genes are regulated. This regulation occurs before transcription and translation because it saves energy. Combinatorial control takes in many signals and decides whether the gene should be on or off. There are enhancers on the DNA which bind to DNA binding factors that have transcription factors attached. These transcription factors relate their message to a mediator complex that drapes over the pre-initiation complex. The mediator complex is made of many proteins that have to work together, or else it will fall apart like a bad move in Jenga. The mediator receives input from the transcription factors, and if more say to repress the gene, then the gene will be off. If more say to activate the gene, then the gene will be on. There are mediators for each cell type, and they can work for different genes in the cell, but they can't work for different cells. They also can't work for housekeeping genes. Housekeeping genes like histones for nucleosomes and proteins the cell always needs can't be regulated. They're always on. We're finally to the last topic. Everything else has been building and growth, so it's only fitting that the last topic is about destruction. RNAi is a system that destroys RNA. Double-stranded RNA, which is normally a virus, is bound and chopped by dicer into small 5 to 20 base pair chunks. These are easily separated into single-stranded RNA. Each of these chunks is called an siRNA, and they are all attached to a RISC enzyme. RISC is an enzyme that can break RNA into individual C, U, A's, and G's. Anything the siRNA can bind to must be a virus because the siRNA came from a virus. siRNA also goes through amplification to create even more siRNA and RISC combos. This mechanism is used as an immune support to destroy viruses in our cells, and it works very well. Nothing is needed to break down the siRNA attached to the RISC because RNA is so fragile it breaks over time. This has been the 8 major topics we covered over the past half semester. I would say I hoped you learned something, but you taught me this, so I hope you enjoyed watching the video.